In this lesson, we look at the structure and functions of the blood vessels. The main blood vessels are arteries, veins, and capillaries. Arteries transport blood from the heart, and each artery divides to form smaller blood vessels known as arterioles. So from the heart, there's a main aorta that divides into various arteries taking blood to different parts of the body, and then each artery divides into arterioles. The arterioles divide further to form the capillaries. Then capillaries join to form venules, and several venules merge into a main vein through which blood is taken back to the heart. So arteries convey blood from the heart, divide into arterioles, then arterioles into several capillaries, then capillaries join to form the venule, and then several venules merge into a vein, and then all the veins from different parts of the body join the major vein, that is the vena cava, and through the vena cava, blood is taken back to the heart. Now, we first start by looking at the structure and function of an artery. The internal structure of an artery reveals that the wall is made up of 3 layers. There is the innermost layer of artery made up of the endothelium tissue that's known as tunica interna. The endothelium is composed of a single layer of cells. Now this layer is found in all blood vessels, not just the artery. It provides a smooth lining which offers the least possible frictional resistance to blood flow. Then the middle layer of the artery is composed of elastic fibers and smooth muscles. It's known as the tunica media. This layer is the one that is responsible for the dilation and constriction functions of the blood vessel. The outermost layer, known as the tunica externa, consists of connective tissues, which are mainly the collagen and elastin fibers. Let's next look at the capillaries. Structurally, capillaries are the simplest part of the blood circulatory system. Even though functionally they form one of the most important parts of the circulatory system, because it is at the capillaries where exchange of substances between the tissues and the blood takes place. Now, each capillary is made up of endothelial cells that are only 1 cell thick. That is the wall. If you look at the cross-section of a capillary, the wall is made up of just 1 layer of cells. This provides the least distance for exchange of substances between the blood and the surrounding tissues. Now, capillaries are numerous and are very close to the tissues in such a way that each cell is near a blood capillary. This is important in promoting efficient and rapid exchange between the blood and the surrounding cells. Then the veins. Structurally, the veins are very similar to the arteries in that the walls are made up of 3 layers. There's the innermost endothelium, the tunica interna. the tunica media made up of smooth muscles and elastic fibers, and the tunica externa made up of connective tissue. However, there are some structural differences between the veins and the arteries, owing to the fact that they carry blood that is under different pressure. One of the very distinct difference is that in major veins, there are semilunar valves that project from the endothelium. So the inner lining of the veins do contain semilunar valves at regular intervals. These semilunar valves prevent backflow of blood because the blood flows under relatively low pressure in the veins. So they need to have the semilunar valves to prevent the backflow of blood. Now, the forward flow of blood in the veins is assisted by contraction of skeletal muscles surrounding these veins. So in addition to the presence of the semilunar valves, the skeletal muscles surrounding the veins also do assist in pushing of the blood towards the heart. Now, functionally, all veins carry deoxygenated blood with fewer nutrients but more nitrogenous waste and other metabolic wastes. Now, except the pulmonary vein that carries oxygenated blood, all the others do carry deoxygenated blood. Now, we can look at the differences between arteries and veins. For convenience, we can look at functional differences and also the structural differences. Functionally, the arteries carries blood that is under considerably high pressure, while veins carry blood that is under low pressure. So the first point of functional difference is the blood pressure. High pressure in the artery, low pressure in the vein. Secondly, and because of the first point, in the vein arteries, the blood flows rapidly and in pulses. Blood flows rapidly and in pulses, but in the veins, blood flows smoothly, so there are no pulsations. And three, another functional difference is that in the arteries, the blood that is transported is oxygenated. The arteries transport oxygenated blood except the pulmonary artery, whereas the veins transport deoxygenated blood except the pulmonary vein, which transports oxygenated blood from the lungs to the heart. Structurally, if you look at the walls of the two, compare the thickness. So arteries walls are thicker and more muscular than the veins. That is a very distinct difference. Thicker walls in the arteries, thinner walls in the vein. This is because of the thicker layer of smooth muscles. Secondly, arteries have narrower lumen compared to that of the vein. Now remember, the veins transport blood back to the heart, and the blood flowing through the vein is under considerable low pressure. So the wide lumen offers minimum resistance to flow. The arteries have a thick wall to withstand the high blood pressure that flows within it. And also the narrow lumen helps to maintain this high pressure. So these structural differences are directly related to their function. Arteries carry blood under considerably high pressure, hence they need the thick wall to withstand the high pressure, the narrow lumen to maintain the high pressure. In the vein, the blood pressure is considerably low, so the wide lumen offers minimum resistance to flow. A third point of difference is the presence of valves in the veins and the absence in the arteries, except at the base of the large arteries leaving the heart. So that is, except for the outer and pulmonary artery that have semilunar valves at the bases, arteries have no valves. Veins, on the other hand, have valves at regular intervals through their length. Again, this is related to their function. They transport blood back to the heart, which is under considerably low pressure. And so the valves help to maintain the forward flow, that is the unidirectional flow of blood from the tissues and toward the heart. So those are the main differences, both functional and structural differences between the arteries and the vein. Now we can look at tissue fluid formation. Let's look at the process that goes on at the capillaries as the blood flows through them. Now, as mentioned earlier, the capillaries provide the site of exchange of substances between the tissues and the blood, and therefore it is important that they are structured and they are able to facilitate this exchange process. Now, the exchange of substances occurs by a process known as ultrafiltration. Ultrafiltration comes about when the blood flowing through the arteriole, which is under high pressure, enters the relatively narrower capillaries. So because of the reduction in the volume, because of the narrowness of the capillaries, pressure builds up. This pressure then forces components of the blood that are small enough to filter out through the capillary walls and into the intercellular spaces. Okay? So the branching of arterioles into capillaries not only increases the surface area for the capillaries and also increases the pressure of blood in them because of the narrowing of the capillaries. And this is what forces molecules that are small enough out of the blood across the capillary walls that, remember, are only one cell thick, and they also do have pores. So the small molecules are forced out into the intercellular spaces. So the molecules that move out and fill the intercellular spaces make up the tissue fluid. This tissue fluid formed is very similar to the plasma of the blood in terms of composition, except that it lacks red blood cells. It does have some white blood cells, and it also lacks large plasma proteins that cannot filter across. However, the tissue fluid contains important substances such as glucose, oxygen, amino acids, vitamins, hormones, and mineral ions that are taken up by the cells either through diffusion or active transport. Now, as a result of the ultrafiltration, there's a drop in the pressure of the blood that remains the capillaries, such that at this end, the venule end of the capillary bed, the blood pressure is less than that of the tissue fluid, because as the continuous ultrafiltration process takes place and more tissue fluid is formed, the pressure within the tissue fluid will be higher than the reduced pressure at the venule end. So some substances in the tissue fluid will then move back into the blood capillaries at the venule end, and these may include waste substances such as carbon dioxide and nitrogenous wastes. Water will also move back into the blood circulatory system by osmosis, because the filtering out of solutes such as glucose and amino acids raises osmotic pressure of the remaining blood. So even water will move by osmosis back at the venule end. Now, excess tissue fluid that cannot seep back at the venule end enters the lymph capillaries that make up the lymph vessel. So the excess tissue fluid enters the lymph vessel and becomes the lymph. And through the lymph vessel, the lymph is taken back to the circulatory system and rejoins the blood. Then the heart pumps the blood again out through the arteries. Arteries divide into arterioles, and then at the capillary bed there is ultrafiltration. With ultrafiltration, nutrients filter out, they are taken up by the cells. The cells release waste products into the tissue fluid. Some of the tissue fluid seeps back into at the venule end and thus rejoin the blood circulatory system, while some of the tissue fluid will filter back into the lymph vessel, which makes up the lymphatic system, and through the lymph vessels, the lymph is taken back to the blood circulatory system, and the process continues on and on.